This lecture series covers the topics in Chapter 9 and 10, which has to do with nucleic acids. We are going to be discussing their structure and their biosynthesis. There are two types of nucleic acids inside cells. The first one is known as ribonucleic acid, and it can be abbreviated into RNA. The next one is deoxyribonucleic acid, and we also know it as DNA. When it comes to the levels of structure for nucleic acids, they are organized in a similar way to what we've seen previously for proteins. The primary structure for nucleic acids is the order of the nitrogen bases in a polynucleotide. Understand that the word polynucleotide is another word that we can use for nucleic acids. The nitrogen bases that are present in DNA and RNA are the following. The nitrogen bases in RNA include cytosine, guanine, adenine, and uracil. Three of the bases that we saw in RNA are present in DNA: cytosine, guanine, and adenine. The difference between the RNA and the DNA nitrogen bases is that in DNA we have a new base that is called thymine. The secondary structure for nucleic acids is the 3D structure of the backbone found in these nucleic acids. In the figure that we have on this slide, that is designated by this thick blue line. So RNA, because it's a single-stranded molecule, has one backbone. DNA, because it is a double-stranded molecule, has two backbones. The tertiary structural level for the structure of nucleic acids is the supercoiling of the molecule. Specifically, when we discuss this topic later in this lecture videos is going to be how supercoiling has to do with how DNA is packed in prokaryotes and eukaryotes. These nucleic acids, or in other words, polynucleotides, are formed by nucleotides. Nucleotides can be defined as the basic unit in nucleic acids. Nucleotides are composed of three components. We have a phosphate group, a sugar motif, and a nitrogenous base. Sometimes you will see that I will be using interchangeably nitrogenous base or nitrogen base. Now we're going to explore more in detail each one of these components. When it comes to the sugar motif that is present in DNA and RNA, understand that we have a ribose sugar present in these two nucleotides. The difference is that in DNA, our ribose sugar is deoxy. And deoxy comes from, if we look at carbon 2 in the ribose motif, carbon number 2 is now going to be a CH2 group, meaning deoxy means that there's a lack of oxygen. Now we lost an OH group, because if we look at the ribose sugar, carbon 2, it is a CHOH. So the absence of that OH group in the deoxyribose is what makes it deoxyribose. Other things to notice when it comes to the sugars is the designation beta and D. They are present in both sugar motifs. Now, the beta designation refers to the position of the hydroxyl group on carbon number 1 in the sugar motif. Because the OH group is pointing up, that's what means that it is in beta position. The D designation on our sugar motif means that the sugar motif shifts polarized light in an instrument called a polarimeter to the right, or in a dextrorotatory way. That's where the designation D. We already explored the different nitrogen bases that are found in DNA and RNA. Now let's look at them in terms of their structure. When it comes to the nitrogen bases, we can separate them into two different classes. Some nitrogenous bases are going to have a pyrimidine ring, and some nitrogenous bases are going to have a purine ring. As you can see, the nitrogenous bases that contain a pyrimidine ring they are going to be cytosine, thymine, and uracil. And I explained a few moments ago how thymine is present in DNA, uracil is present in RNA, and if we look at their structures, the difference between them is that methyl group on the left side of the molecule. When it comes to the nitrogenous bases that contain a purine ring, they are only adenine and guanine. One of the features or the chemical features that you should know about these different types of rings is that both rings, if we classify them in terms of the functional groups, are aromatic, heterocyclic amines. Because they have those chemical features in them, nitrogenous bases are hydrophobic. And later in the lecture series, we're going to be observing how that influences the structure that these nucleic acids adopt. When it comes to having the components of nitrogenous base, sugar motif, and phosphate, we have different structures that can be formed, starting with a nucleoside. A nucleoside is going to be when we have a sugar motif and a nitrogenous base linked together. Understand that these two components of these structures are going to be bonded by a specific type of bond. In the case of a nucleoside, which has the components of nitrogenous base and sugar, that nitrogenous base is going to be bonded to the sugar motif at carbon 1 through what is called a glycosidic bond. And glycosidic just means sugar. So that's why, since we have sugar binding a nitrogenous base, that is called a glycosidic bond. At times you will see that they also call it an N or beta N glycosidic bond. The beta is, again, because carbon 1 is pointing up, and in this case, because of the chemistries, the hydroxyl group left. N is because we're bonded to nitrogen, and glycosidic because we're bonded to a sugar. Understand, nucleosides do not have phosphates, because if phosphate is present on a nucleoside, then it's going to have a different name. And those are known as nucleotides. So when it comes to nucleotides, as I just expressed, is when we have a nitrogenous base bonded to a sugar motif, and then we have a phosphate group. Understand that this phosphate group bonds to the oxygen atom on the 5th carbon on our ribose or deoxyribose sugar motif. So the oxygen group on carbon 5 in our sugar motif is what gets phosphorylated. Now, let's explore the nomenclature when it comes to how we define talking about just nitrogenous bases versus nucleosides versus nucleotides. So the second column in this table express what are going to be the names of our nitrogenous bases, which we introduced previously. So as you can see, the nitrogenous bases for RNA and DNA, adenine, guanine, and cytosine is going to be present on both. But in the case of RNA, uracil is present, and in the case of DNA, thymine is present. Now, when it comes to what is the name, if that nitrogenous base is bonded to sugar, then understand that the name changes. Now, I will suggest that you just memorize those names because there is not a systematic way of naming when it comes to naming nucleosides. So, understand that when it comes to this, your adenine turns into adenosine. Your guanine turns into guanosine. Cytosine turns into cytidine. Uracil turns into uridine. And thymine turns into deoxythymidine, because remember, the sugar that thymine is bonded to is a deoxyribose. So a thymidine needs to have the word deoxy because of the sugar motif that is present or bond to it in the case of a nucleoside. So that's why as we see the adenosine, guanosine, and cytidine in DNA, they all have the word deoxy. Now, one of the things that I want you guys to notice is that we also have a 1-letter code for bases, the nitrogenous bases and the nucleosides, and they are the same. So as you can see, the letter A is going to be for adenine or adenosine. The letter G is going to be for guanine or guanosine. The letter C is for cytosine or cytidine. U for uracil or uridine, and T for thymine or deoxythymidine. Now, understand that this is, as I just mentioned, things that you guys should know and memorize because there's no systematic way to or rules for naming. Now, when it comes to nucleotides, the difference between the names in nucleotides versus nucleosides is that at the end of the name, we're going to add how many phosphates we have in those structures. And as you can see in the table on the 4th column, okay, we have only monophosphates. So when it comes to the nucleotides, the last word represents the number of phosphates in the nucleotide structure. So what does this mean? That in this case, monophosphate represents one phosphate linked to the oxygen on carbon 5 in your sugar motif. We can have up to 3 phosphates present in these molecules. So we can have a diphosphate or a triphosphate. Diphosphate means that we have 2 phosphates present in the molecule. Tri means 3 phosphates. When it comes to the abbreviation for nucleotides, understand that it is given by the 1-letter code, okay, of your nucleoside name. Remember, nucleoside name I imply because there's base and sugar, but we also need to include the phosphate. And then the first letter in your prefix for how many phosphates you have, and the P for the phosphate. So AMP stands for adenosine monophosphate. So if you had, for example, ADP, then that means that is adenosine diphosphate. So we have the A for adenosine, the D for the di, and the P for the phosphate. That's where ADP comes from. When it comes to nucleotides, understand that they have functions that extend from the synthesis of nucleic acids. So for example, FAD, NAD, and coenzyme A, we're going to see them performing biochemical reactions. Specifically, we see them involved in oxidation reduction reactions. ATP, even though it is also utilized to synthesize nucleic acids, is the energy currency of the cell. And once we get to metabolism, we will see this in action. Now, one of the things that we need to explore is how these nucleotides are going to be reacting to give nucleic acids. When it comes to this, understand that we need to envision a nucleophilic attack on specifically an electrophile in order to create these bonds. So in this reaction, what happens is that if we highlight the atoms, at carbon 3 in this case, deoxyribose sugar, we have a hydroxyl group that is going to be acting as a nucleophile. This nucleophile is going to be reacting with the phosphate group, okay, in the incoming nucleotide base. And when it comes to the phosphate, we can label them as alpha, beta, and gamma. And specifically, whenever we have these reactions occurring, we are going to have a triphosphate. And the first phosphate is the one that is going to be acting as an electrophile. Once we have that 3' hydroxyl attack that 5' phosphate, okay, then what we're going to see is generate inorganic phosphate, which means that this inorganic phosphate, which I'm going to highlight in neon green, represents these two groups that are leaving the system, okay? And now what results is that we have a covalent bond that is formed between the 3' carbon, okay, specifically at the oxygen atom, bonded to the phosphate that it is present in the 5' carbon, okay, in the sugar motif that is, or the nucleotide that is entering the system. So we're going to see that when we are synthesizing these nucleic acids, the term 3' to 5' refers to the nucleophile, which is at the 3' position. Remember, that's the hydroxyl group that is going to be acting as a nucleophile, and it's going to be bonded to the 5' phosphate, which is the first phosphate in a triphosphate nucleotide, and the resulting functional group is going to be a phosphodiester bond.